Okay, so I would like to welcome everybody to this webinar, to this transplant webinar. And today our guest is Professor Daninele Campa. He's an associate professor of genetics in University of Pisa, and he will give us a talk about modeling pancreatic cancer risk with Omics data. So it is a pleasure for us to hear you, Daninele.
Thank you very much, Ingrid. Shall I share the screen? Okay. You should be able to see and hear me, right? Right, thank you. So thank you very much. Good afternoon, everyone. I see in the audience many faces, many people that it's a long time that I don't see in person. So at least here. So what will I talk about? Well, I am a geneticist, so I will talk about genetic susceptibility to pancreatic cancer. And we will see if there are individual genetic variants that can predict PDAC development, and if and how we can use germline genetic variability to model pancreatic cancer risk. And important question as well, how can we integrate germline genetics with other omics data? And if there is a target group of individuals in which we should use omics data to with prediction models. So we all know these statistics. Is the first time here that I use the statistic which were updated in 2022. Last talk that I gave, they were the one on 2020, but basically there are no differences here. On the left, you can see the incidence and mortality for around the world for 100,000 individuals. And you can see the pancreatic cancer is the very bottom of this group of high incidence tumors, and that mortality, which is the red one, is basically the same as incidence. Whereas for many other cancer types, there is a very large gap. For example, if you see thyroid, there is the difference between mortality and incidence is striking. And not just that, but this trend is not going away, because if you look at the right-hand side of the slide, you can see that pancreatic cancer mortality is on the rise. And it's very few cancer types that are for which the mortality is on the rise. One is pancreatic cancer as well, and the other is liver cancer. Why? Well, there are obviously many reasons. One of the reasons is the fact that we are unable to predict it. And we are unable to predict it because in large part we actually don't really know what causes it. There are several risk factors, but they are not really strong compared to other cancer types. And can genetics help us? Well, to speak about that, I have to speak about peas and the color of peas. And why? Because do you know to explain the color of a pea? I guess you can. And do you know how to explain the height of people? And of course, the answer to the question is the fact that the color of the pea is explained by Mendelian genetics. There is one high penetrance mutation that explains basically the phenotype. If you have one allele, you are green. If you have the other allele, you are yellow. The environment has nothing to do with determination of the phenotype. And this is called a Mendelian trait, as you know. Just for your curiosity, the mutation that changes the color of the pea is actually the integration of a transposon. How do you explain height in people? Well, we are at a complete different end of the spectrum. To explain height in people, you have more than 12,000 variants, each of which explain just a little bit of the variability. There is of the variance, actually, if you want to use the correct words. So there are 12,000 SNPs in the genome, and each of those SNP has one allele that gets you a little bit taller or a little bit shorter than the average population. It's the combination of those 12,000 variants that explain a large proportion of the phenotype. Of course, with height, with all complex diseases, there is not just genetics, but there is also the environment. So Mendelian and non-Mendelian genetics. Why this was important? Well, because if you want to study pancreatic cancer, we need to know if we are looking at a phenotype which is closer to the color of a pea or closer to height in people. And from a genetic point of view, PDAC is a complex trait. There are several low penetrance variants, some middle penetrance variants, and of course, environment plays a role. This is an old figure now. It has 10 years, but explains, and you can apply this to basically all diseases that have a genetic component, which means basically all diseases. And here you can see on the y-axis the effect of the mutation or the variant. The highest it is, the stronger the effect is. So here we have the red ones with an effect of 10. 10, it's a very, very large effect. If you compare to environmental variables, for example, smoking and lung cancer, the same effect you have if you smoke 20 cigarettes a day and for many years. On the other part, on the left-hand side, we have common low genetic variants, which explain a little bit of the variability of the trait, of the variance of the trait. And the main difference between these two is that rare penetrance variants are really rare. So they are found in very few individuals, while common penetrant variants explain individually not much, but they are very spread in the population. Some have a frequency of 0.5, which means that basically 1 person out of 2 has the risk allele. What do we know about the rare high penetrance variants in pancreatic cancer? We know some. There are several loci and not many. And as you can see, the prevalence is not super high. It's relatively high in familial pancreatic cancer, but it's quite low in sporadic pancreatic cancer. So there is not really much that we can go on if we really want to predict the disease. And these were found with sequencing studies, especially in families. If you want to know more about hyperincidence mutation, this is a review that we have published in Transparent this year, and you can have a more in-depth discussion. You can read a more in-depth discussion about BRCA1 and BRCA2 mutation, for example. What about so to reply to the first question, so to the second question, are there individual genetic variants that can predict pancreatic cancer development? And the answer is no. No, PDAC is not a Mendelian disease. It does not exist, the pancreatic cancer gene. It will never exist. We will never find it. What about the low penetrance mutations, the ones that are the blue dots there? Well, if high rare variants with high penetrance, we have identified them through sequencing, especially sequencing in families or in high selected groups. For common low penetrance variants, we need populations. And we need to study basically all the genome. We have to interrogate all the genetic variability in each of us. And for doing so, we need a technique which is reliable, a technique that is cheap, and a technique that allows us the high throughput in the genotyping. And by far, the most cost-effective technique that we can use, that has been used in the last 10 years, are genome-wide association studies, or GWAS. This is a slide taken by the National Human Genome Research Institute. They have a series of slides for teaching, and I use them very often in my courses at university. And here it explains very well what I'm trying to say. So here you have two groups, individuals with disease, individuals without disease, and you genotype using a chip. This thing here which are closer to the arrows, are cheap. And here you can genotype from 500,000 to 5 million SNPs in one go. And you have two populations here, the brown one and the blue one, and several SNPs. Of course, not all the 5 millions, because that would be impossible, but there are three as an example. The first one, you see that the variants are the green dots. And you can see in the two populations, in the brown and the blue ones, that the dots are the same. They are not in the same people, of course, but the number is the same. So basically, the risk variant is distributed among the two populations without any difference. If you pick up one at random, considering the cases and controls together, one green dot, you have no idea to where to assign the person. There is no different probability in assigning the person in the blue or in the brown population. The same is true for SNP2. Here the dots are yellow, but it's the same. They are distributed in equal number between the cases and the controls. You pick up a yellow allele and you have no idea if the person that you have selected belongs to cases and controls. SNP3 is very different. The red dots are the risk alleles. As you can see, the brown population is enriched by risk alleles, while the blue population has not many of them, has just 4. So if you pretend to have a bowl or a bottle or a barrel in which all these people live together, and you pick one at random, and the one that you pick is red, you have a very high probability that that person is in the brown population and not in the blue one. So the SNP is associated to the disease. And here is what we know. We know around 30 loci which are associated to pancreatic cancer. They are scattered across all our genome. There are a little bit more in the region on chromosome 9, but as you can see, there are several. Some are European specific, some are Asian specific, and some are both true in Asians and in Europeans. Again, here, if you want to know a little bit more about the genetic variability and pancreatic cancer, so the pancreatic cancer genetic susceptibility, here there are a couple of reviews that we have done in Transpan, and another one that was like kind of celebration of Pandora, which is another consortium that I belong to. Now, what are the characteristics of these SNPs associated to complex diseases, pancreatic cancer included? They have two characteristics which are striking. First one, you have not to look at the column with the dotted line, but the one on the right, sorry, the p-value. So we have very, very low p-value for these associations, meaning that we are sure that the variants, the SNPs, are associated with the disease. The second one are the odds ratios, so the strength of an association. The higher it is the odds ratio, the stronger is the association. The lower is the odds ratio, and lower mean closer to 1 is the odds ratio. The weakest is the association. So you can see here that all the pancreatic cancer susceptibility SNPs have very low p-values and very low odds ratios. So they are all associated with small effect. Just to be sure, an odds ratio 1.46, as in the example, means that people carrying the variant allele, the A allele, have a plus 46% probability of developing pancreatic cancer if compared with the C allele. 46%, sorry, it's very low, because this is a relative risk. It's not really relative risk, but with pancreatic cancer, which is a rare disease, basically is the same. So, and they're all like this. All SNPs generally have a very small effect, but this is true for all diseases. So like it is for height, that's why I showed you height. You cannot think to disentangle the effect of genetic variants in a complex disease. You will never think to explain height of people with one mutation. So why should you think to explain the risk of a complex disease with one mutation or with few mutations? It's really counterintuitive. It does not make any sense. What we really need to look, is the combination of alleles. We have to combine the effects. And through the years, after our genome was sequenced, after the first GWAS were out, and after we saw that what we got from GWAS were very, very, very low p-values, usually associations that were confirmed across countries, across ethnicities, across studies, with very low odds ratios, we had to think of ways to combine this effect. And one of these ways is polygenic risk score. Here you see there are protocols now that you can use where to understand that guide you from the beginning to the end, more or less, on how to run a genome-wide association study and how to combine and compute a polygenic risk score. Just as an example, here you have an histogram. These are real data taken from a study that we have carried out in Pandora. Here you see that the histograms represent the number of risk alleles. And the two colors represent the controls and the cases. So red people, the red histograms are cases, the blue ones are controls. You see here that there is a almost Gaussian distribution, and that if you look at the histograms, the highest it is the histogram, the more risk alleles there are. If you look, and sorry, that's the percentage of people. If you look at the x axis, you see the number of risk alleles. So you start with, theoretically, if you have 30 SNPs, and every single SNP can have 3 genotypes. You can have genotype 0, which you have 0 risk allele, genotype 1 for the heterozygous, in which you have 1 risk allele, and genotype 2, in which you have 2 risk alleles. So theoretically, the distribution of risk alleles can vary from 0, if there is a person that is homozygous for all the protective alleles, and 60 for persons that are homozygous for all the risk alleles. In reality, the distribution goes from, at least in our study, which was done with approximately 8,000 individuals, so, well, not millions, but still, it's a good number. You can see that the distribution goes from 17 to 40. If you look at the 17, 18, 19, 20, you see that the majority of the people are controls. And so the majority of people that have a very low number of risk alleles are all controls. The majority, on the other hand, the majority of people that have a high risk allele score are instead all cases. So cases and controls are divided by risk alleles. So why I said size matters? Well, what's going on? Before we talked about peas, and now we talk about sheep. Well, it's really difficult to make jokes when at the monitor, because I don't know your faces. I hope that some of you at least is smiling. But if I want to look at the phenotype that is not heterogeneous, like black or white here, it's very easy to identify individuals which are different. For example, this guy. And if the effect of the variant is very strong, it determines the phenotype entirely. It's very easy to identify it. But in a complex disease, it's not like so. And what really matters is the combination of risk alleles. So that's why we need large numbers, because we need to identify individuals that have a combination of a high percentage of risk alleles. So basically, we are interested in this part of the distribution, only the ones with high risk alleles. And this can be a very small percentage of the population. And therefore, size matters, because we need a large sample to identify all the clinical relevant ones if we don't know them, or to compute a polygenic risk score if we know which one we have to combine, but we need to find people that have the correct number of risk alleles, a high enough number of risk alleles. Will it work? Well, yes, it will, at least in some extent. This is an example of a paper that was published in March this year, and it was done in UK in a cancer which is really different from pancreatic cancer. It's prostate cancer. You know that there is a screening for prostate cancer which is done with the prostate-specific antigen, PSA. This is used. This is really used. I went to, for example, to San Raffaele 1 month ago to speak with a friend, and when you enter, San Raffaele is a large university hospital in Italy. When you enter in the hall, there are movies in which they explain to you several things. It's really well done, and there was also the explanation of what PSA is. So you should do it, why you shouldn't, which age you should do. So it's something that it's done, and the general population knows about PSA. But it has a high rate of false positive. This idea of this study was to compare a polygenic risk score, something that nobody ever done in real clinical setting, with something that is currently used in clinical setting for a common cancer. So they invited 40,000 individuals in the UK, and they investigated a polygenic risk score that was computed with 130 variants. They identified 745, which is a little bit more than 11% of the population, so that's why we need to look at the right-hand side of the distribution, that had a polygenic risk score in the 90th percentile or higher. Those people, those 40,000 people, they did not know if they had pancreatic cancer or not. Prostate cancer or not. They were people of the general population. They invited this person to have a proper screening, a real one, and they detected prostate cancer in 187. So 40% of the people in the 90th percentile of the polygenic risk score actually had prostate cancer, and they did not know about it. And this outperformed gloriously PSA. Can we do that for pancreatic cancer? Well, let's keep that question for a second. And let's go on another question which is important as well. Can we integrate germline genetics with other omics? If so, which one should we choose? Well, all of them. There is no reason to leave one out, including the exposome. The exposome is important in pancreatic cancer. Here, it's a study that we have done, and using a UK biobank, we have analyzed around 1,500 exposome variables, so variables associated with lifestyle, with your diet, with environmental pollutant, with what is your work on, what you work with your parents, a detailed questionnaire in the UK biobank. As for GWAS, on the y-axis you see the p-value. The highest it is, the lowest the p-value is. It does not mean that the association is strong. It just means that the association is statistically sound. And as you can see, there are several things that we knew already. For example, obviously we knew that pancreatitis is a risk factor for pancreatic cancer, that smoking is a risk factor for pancreatic cancer, and here you can see that they are on the top of this list as for p-value, not the strength. Whole body fat-free mass was also associated, and here you see on the right-hand side of the presentation, I can use the laser. Here we have a high PRS, so a high polygenic risk score, which is good because we are geneticists, so we like to have a polygenic risk score usable for detection. How we can integrate germline genetics with other omics? Well, there are several ways, but of course one that is getting a lot of attention are artificial intelligence models. So this is another study that we have done. We use, again, UK Biobank. We use several variables, exosome, PRS, use a train and test set. We use several machine learning models, and we also used, as you can see here, an explainer. What is an explainer? Well, machine learning models, what they do is that they give you a prediction, and you have several parameters which you can test the performance of the models, and they give you, okay, the model explains with specificity or sensibility and other parameters the model that you have, the data set that you have selected. But then you need something to understand what you actually are getting from those data. And those are called explainers. There are several, and basically what they do is that they try to give a ranking on the importance of a variable that is called feature, but it's the same, or they try to build rules in the model itself, like saying if you are more than 59 and you smoke 29 cigarettes a day and you have pancreatitis, then this happens. So one which is used by the majority of the study is SHAP. Sorry, I hear echo. Probably there is somebody with the... Can you maybe close? Okay, now it works. Right, so can we use these models to predict PDAC? Not PDAR, sorry. Well, actually, no, we cannot, because PDAC is too rare. So even if we end up with a very specific, very sensitive model, we will have to analyze thousands of, fortunately, we will have to analyze thousands of individuals to identify a very small number, based just of the incidence, which is around the world of 17 in 100,000. If we screen 1,000 individuals, we will find 17 new pancreatic cancer cases. This is obviously an oversimplification, but to give you an idea. So what should we do? We should focus on high-risk individuals. And who are high-risk individuals? Individuals in high-risk families, individuals with hereditary pancreatitis, individuals with late-onset diabetes, and individuals that have precursor lesions of PDAC. One of the most common found precursor lesions, at least from a patient way, if you look in the population, you screen the population, not just PDAC, but the population, one of the most common are IPMNs, intrapancreatic mucinous neoplasms, and we have done a study with several omics and the integration of several omics, so I'll show you some preliminary results of that, and to understand which of these IPMNs, which are cysts in the pancreas, will develop pancreatic cancer and which not. So we used genomic data, epigenetic data, microRNA expression, the exposome, the microbiome, and the idea is to put all this data together and analyze them in a model with a multifactorial risk score using standard statistical methodology and using explainable AI. XAI stands for explainable AI. So using AI and then an explainer. A little bit more about what we are doing. We have more or less 650 subjects for which we have exposome data. We have 450 subjects for which we have NGS exome sequencing data. We have around 100 subjects for which we have somatic exome sequencing data. Then we have dense SNP arrays, so chip arrays, the GWAS, the one that was showing before, with for 650 subjects. And we have the MIRNOM for around 350 subjects. And we are doing oral microbiome short-rank sequencing, and we more or less have 40 subjects. We are starting to accumulate all these omics. We have the metabolome for 100 subjects and the whole genome sequencing done with Oxford Nanopore for just 2 subjects, the first 2. And because we are setting up the technique here in this month, so it's going a little bit slow. And we have the epigenome for 2 subjects done with Oxford Nanopore, but we are in the process of analyzing and producing more data. This actually is a picture that I sent on a grant application. So, but basically the idea is the same. We take all the data, we combine them with a machine learning model, and we want to see if we are able to define individual IPMN patient that will progress into pancreatic cancer or high-grade dysplasia, or individuals that will not. These are very preliminary data because, as you've seen, the data set is not filled. For some variables, we have almost all the subjects that we have in the study. For some variables, we have less than half, and so on and so forth. And here, we have done several tries using only clinical data, using data and the polygenic risk score, or using the polygenic risk score, so the common variant and also some rare mutations, and then all the omics that we had. So basically, if you go from left to right, the number of individuals decreases. Here on the first column, you have parameters in which you score the models, and just to be simple, you can consider precision and recall as sensitivity and specificity. They are not exactly the same, but almost. So you want to have a high number, the closest to 1 as possible. So if we use only clinical data, although the number of individuals that we used is the highest possible, the results, the performance of the model is not great. If we add the PHS, it immediately gets better. We have, for example, precision for IPMN with progression goes from 0.35 to 0.67, and recall for progression that remains the same, but we have also recall for non-progression that goes to 0.91, which in IPMN is very important because it would be very important so to say to a patient, you don't need to worry. You will the chances of you getting a pancreatic cancer from your IPMN are really low. Adding together all the genetic data that we could, the model gets better. It gets much better if we put all the omics. Now it begins to look like something which is very promising. Of course, all the omics limits the samples that we used here around a little bit less than 300, but anyway. So the feature importance here is what SHAP normally gives you, and this is the very last slide, so don't worry, I'm done. And here is how you read this figure. You have the one on the right. You see that there are dots, and that there is a line in the middle in which there is written 0. If the dots are on the right part, it means that increases the risk of being of describing or predicting the outcome. And if it is red or blue, it means that the value of the variable is getting higher with red and lower than blue. So for example, if you have a dot which is on the red, a very red dot on the right hand side means that the increasing the value increases the risk. While if you have a blue value on the right hand side means that decreasing the value of the various variable or that variable that you're looking for decreases the risk. For example, if it was smoking, decreasing the number of cigarettes, so blue dots will decrease the effect. But here you can see, and also ranks the variables for their importance. And the most important one was the polygenic risk score. You can see that basically all the red dots are on the right-hand side and all the blue dots on the left-hand side, meaning that the polygenic risk score alone is able to discriminate very well these two groups. Second place is one Myrna. Then we have other Myrnas, ABO blood group, which was also the model identified as important. Of course, here there are bias in this estimation. It's just a preliminary thing that we did because clearly we investigated very thoroughly the genome and not really thoroughly all the other omics. So clearly they seem to be most important because I was done. So in conclusion, predicting pancreatic cancer in the general population is going to be difficult because it's rare, but multi-omics integration instead will be invaluable in high-risk individuals to stratify them and make meaningful prediction. IPMNs were just an example, but other subgroups can be used and should be used, in my opinion. And also, these results, even if preliminary, show that genomics is a powerful tool in prediction and should be integrated with other omics. And with this, I would like to thank everybody and for your patience. Thank you very much.